Hi, good afternoon, and welcome to another episode of the Botany Bistro, coming at you live from the Civic Garden Center in Cincinnati, Ohio. My name's Mary Dudley. I'm a botanist, and I started this little series to give you some bite-sized botany that you can use to improve your at-home gardening, and any other type of growing you like to do, and just your appreciation of plants in general, since they are so fascinating and there's so many cool things to find out about them. So today actually is our 14th episode of the Bistro. Pretty exciting. We're going to be talking about plant breeding and genetics today. All of our episodes are archived on YouTube and at our website if you ever want to learn more about that. The Civic Garden Center has been here since 1942, helping people with their plant questions and gardening journeys. We really got a kickstart with the Victory Garden movement, and urban agriculture is more important than ever. So if you're interested in starting that journey, please keep us in mind as a resource. If it's your first time, I will give about a 30-minute, 40-minute chat, and then I'll open up the floor for questions, and so you'll be able to turn on your microphones and send some messages to me if you have any questions. And as always, I'm happy to answer your questions anytime, and you can contact me through the Civic Garden Center. As you can see, the weather is changing. Happy December, everyone. So the beautiful red bud behind me has lost all of her leaves. She does have quite a few seed pods left, and we'll be looking forward to those buds coming back. But in this restful season, I hope that you're all healthy and enjoying the months ahead. So today what we're going to do on the Bistro is we're going to really get into plant breeding and genetics. This is actually going to be a 3-part series that we'll do. And so there's lots to cover, and so I've broken it up into different groups. And so we'll be talking a little bit about the beginnings of plant breeding, some basics of genetics, and then we'll go on from there to session 2 and session 3 over the next month so that we can really dive into some of the current methods in plant breeding and some things that you can try out at home as well. So just a recap on some of the other things that we have done. We've talked about plant pathology in our most recent sessions. That was really useful. We had a special guest on last time who talked to us about some gardening pathogens that you can avoid, hopefully this coming season. And we've talked about plant physiology, systematics, anatomy, morphology. There's just so much in the field of botany. And so we've really broken all these down for you, and we're really excited to continue the series. Today we're going to have some Mexican corn casserole for lunch. So I always try to have a lunch that is relevant to the topic of the day. So you'll have to stay tuned to find out why I decided to choose this casserole for lunch. It smells very good, and it's perfect for cold weather. We're going to learn a little bit about corn, sunflowers, and poinsettias today. So you can kind of scratch your head a little bit now and think about how those things might all be interrelated. We always have a little bit of homework we encourage you to do during the week off that we have in between, since we're on every other Friday. And so your homework is going to be to tell us about a unique plant that you really like. And there's probably a pretty good chance that some of the more variegated or unique houseplants that we have may have been the result of some breeding programs, even some of our favorite foods. So we'll discover that over the next few sessions. But let's get into it. So plant breeding, what is it? Well, the word breeding probably brings to mind the replication of something and how you kind of get something to continue on in its life cycle. And so plant breeding uses principles from several different sciences to improve the genetic potential of plants. So in order to really build on some of our modern plant breeding programs, scientists had to get a very good foundational understanding of genetics and how genetics works in the plant world. So that was a very important first step. And so we'll kind of start there along with our predecessors, and we'll discover a little bit more about genetics. But these plants are then improved for different uses. So how can we make food that's more nutritious? How can we create plants that can grow in drought tolerant conditions? But also, how can we create plants that are good for feed for the animals in our lives, not just our house pets, but also the animals that live on farms. And so the other portion of plants that we use a lot are going to be fiber, fuel, and shelter. And so we're always thinking about how we can better use the plants in our lives and make sure that they're healthy and that we're being efficient. And so especially in the face of the climate crisis that we're in right now, it's important for us to think about ways that we can modify or just encourage traits in plants that will make sure that they are more sustainable. We have less inputs that we're putting on them throughout their life cycle. So it's a science-driven process, typically resulting in new plant varieties. And these plant varieties can go by a variety of different names as well. But we're trying to just build upon what existing genetic material is there and going forward with that. Now, I do want to make clear that these topics are not going to go too deep into biotechnology, since I already have a couple of series dedicated to biotechnology coming up in the next few months. So we are going to talk about more traditional plant breeding methods today and in the next couple of weeks. But stay tuned for those biotechnology pieces because it is very fascinating what we're doing currently. And we just won't be able to have time to go super far into that today. And so how long have we been doing this? Well, a long time. So the science of plant breeding really has been going on for about 10,000 years at least. This is just what we know about our human history. And it was about 8,000 BC when Mesoamericans were working to domesticate corn. Bingo! So we already hit the choice for our lunch today. Has some corn in it. It's a crop that has been modified and selected for and bred for thousands of years. And so we use a lot of it in many different ways. You might be surprised about all the ways that we use corn in our lives. But it is a product that is totally different from its original ancestors. So we'll go into that as well. And also there's evidence that about 700 BC, Assyrians and Babylonians were hand-pollinating certain palm species in order to make them more efficient and more robust. So we have this history of early farmers, when we started to settle down onto farms and trying to make all of the elements that we were growing on those farms very productive, using something called selection. So that's a pretty simple, I don't want to oversimplify. I mean, it still takes a lot of technique, but you are identifying traits that you would like to have in your plants, and you are saving that seed. And when you have something that is not desirable, you remove that or cull that, C-U-L-L is that. And so it's really just being picky about what you have in your garden and in your crops. And so as these early farmers were, really paying attention to what they were growing and starting to remove the things they didn't want and continue to select for what they did want, they were able to see these different species change over time. But let's back up a little bit. So even though our early ancestors may not have known on the molecular level what was happening, we have been able to build really strong plant breeding programs that are very specific and targeted based on our understanding of genetics. So let's think a little bit about what genetics means. And so in the plant world, Gregor Mendel is a name that you'll hear quite often, considered the father of plant genetics. And he was experimenting on pea plants. So just our little garden peas. And so he wanted to, kind of figure out more about why did some of them have different seed color and some of them have different bloom color. Some of them grew to different heights. And so he was doing a lot of different experiments with that. He was working on this in the 1860s, and he put together the principles of inheritance based on his experiments. So, for example, He worked with 7 different characteristics of plants, and let's just focus on seed color. So he had a true breeding yellow parent and a true breeding green. So these plants would produce those seeds reliably. This parent was always producing yellow seeds. This parent was always producing green seeds. And so when they were combined, and he hybridized those, so basically he's taking pollen from one and pollinating the other, and then he's collecting that seed and he's growing that plant again, and then he's harvesting those seeds. So when he had that first generation, he noticed that all of their offspring produced yellow seeds. Even if they, were, even with the pollen coming from the parent that was producing green seeds. So he's like, why is that happening? So that first generation is all yellow. The next generation, however, had green peas in a ratio of 1 green to 3 yellow. So now he's like, oh, wait. So he grew a second generation from that original hybrid, and now he's starting to have some variation. And so he was able to coin the terms dominant and recessive. He didn't really know what was happening. He called them invisible factors. We now call those genes. But the traits in an organism are predicted by genes. So he was just starting on that journey and trying to identify what are these invisible factors. And so now I want you to kind of put into your head the good old Punnett square we all remember from high school, probably some of us. And so it's basically like a window pane, four panes in the window. And so you have a trait that is controlled by genes, and for him they were these invisible factors, but you can express those by different alleles. And so at the top of our 4 window pane we have an uppercase B and a lowercase b, and then on the side maybe we have an uppercase B and a lowercase b. And so then when we have this 4-pane window, 2 of these boxes are going to have the same. It's going to be big B, and then other ones little b. And then 2 of the boxes are going to have the variation, little b and big B, little b and big B. And so, okay, what's happening here? So 3 of the boxes have big B's in them. 1 of the boxes has just 2 little b's. And so what he determined from these experiments was that this recessive expression of the green color of the seed was only going to be expressed if the dominant was not present. And he was right about that. And so this was a pretty exciting time. He was actually not recognized for any of the work during his lifetime. It took decades after for people to rediscover his work and be like, oh my gosh, like this guy was getting it. And so it's really nice now that we can recognize all of that labor that he put into it. But he formed these principles of inheritance. And the first one is the law of segregation, which says that each inherited trait is defined by a gene pair, and that parental genes are randomly separated into sex cells so that they only contain 1 gene of the pair. So then the offspring inherit 1 genetic allele from one parent and 1 from the other, and then they unite during fertilization. So that's the first law. The second one is the law of independent assortment, which means genes for different traits are sorted separately from one another, so that the inheritance of one trait is not dependent on the inheritance of another. And the third is the law of dominance, which means an organism with alternate forms of a gene will express the form that is dominant. And so we can, kind of apply this to other things that we have seen in life. And so when we have a dominant allele, it's going to be the one that we see. And so he figured out that this yellow seed was dominant and the green was recessive. Now, I do have to say that not all traits are inherited in this way. It's very simplified. It was neat because his experiments really showed that some of the characteristics he was studying were pretty easy to line up in this way, and some of them were not. And so there are other types of inheritance that do not follow these Mendelian laws, but some of them do. So it's a really good place for us to start. Now, I didn't back all the way up to talk about the difference between meiosis and mitosis. And so if you're not sure about how sex cells are able to only have one half of the genetic material, that's pretty interesting. So if you want to look up the difference between mitosis, which is cellular reproduction that duplicates a cell, versus meiosis, which is where it only contains half of the material so that it is ready to be paired with another cell during fertilization. So we go through these processes in our own biology, in our bodies, and plants do this too, because in their pollen grains, they have cells that have been undergone meiosis so that they only have half the amount of genetic material, so they can be paired up with something that is in the ovaries of the plants. So yeah, I guess maybe I'll do another session where I really back up on some of that. But that does go into plant physiology, and so if you're interested in reviewing some of those sessions, I encourage that. But yeah, understanding kind of, okay, what are these invisible factors, and how can we leverage this information to create new species that are going to be showcasing the desirable things that we want. And so this whole process of selective breeding, you have to be very controlled. You don't want any pollen that you are floating around, really. So a lot of times you're bagging things, maybe you're even removing flowers at certain times, you're hand pollinating. It can be very labor intensive. Also, you are then thinking about how you're going to grow the next generation. And so you're constantly kind of redoing your experiment over and over and over. This takes a long, long time to be able to grow up a parent population and then make a hybrid and then grow the offspring of those hybrids. Peas don't take too long, but there are some plants that could take a really long time to do this. And so they have shortened the process using different technologies. So we're going to talk about that just a little bit. There's a couple other cool stepping stones, milestones in the world of genetics that we want to discuss, and just breeding types. So in 1903, it was, became pretty obvious that the phenotype doesn't always, predict the genotype. And so genotype is going to be those genes, that DNA that we're looking at. And that was harder to do before microscopy and some of the other tools that we have now. Phenotype is the physical appearance of the plant. And so even though some of us were like, I really want a plant that is shorter. Okay, great. Well, let's take these short plants over here and they'll be short, right? Well, not necessarily. There are environmental factors that influence a phenotype of a plant. So if that plant is growing in poor soil or in a shaded environment, then it might not reach its full stature. Versus when you grow it in an open space with fertile soil, it might be huge. So you couldn't always predict that was going to come true for the plant just based on phenotype. And so that was a big step to be like, oh my goodness, let's think about environmental factors, but really what we're trying to control here with plant breeding is the genotype, so that we have something that's predictably going to follow the rules, should we say, that we have set out for it. We also learned quite a bit about hybrid vigor, which is also known as heterosis. And, we could probably line this up with some of our own human experiments in just our ancient royal families sometimes have, kind of kept it to the family, let's keep it in the family. And it's shown also in plants that inbred lines show deterioration over time and just less, robust. They're not as strong. And so they started hybridizing these inbred lines of different kinds of corn that they were growing, different kinds of beans that they were growing. And they found that next generation, when they were hybridized, was much stronger. And so I love that this is happening, in the early 1900s, but it's something that we are still, kind of teaching about today. Is genetic diversity is so important. And I talked about some of the tragedies that have happened around the world a couple weeks ago with plant pathogens that took over a crop that didn't have high genetic diversity. They were relatively, uniform. And so we really need to protect this genetic diversity in order to save ourselves some hassle and build that resilience up. And so in the 1990s, geneticists and breeders since the 1990s, we've been using molecular markers, DNA-based markers. And so they're able to actually look at specific regions of the plant's genome that are going to influence a desired trait. So instead of just kind of guesswork and all of these subsequent generations and all of the time spent growing out all these plants, we can now use these markers to obtain a DNA fingerprint of a particular variety and develop DNA maps to sequence these genomes. The applications of these markers are used in plant breeding to select progenies of breeding crosses, so particular parent individuals that are desirable because they have the greatest number of desired traits. And so that's pretty exciting stuff right there. The first complete genome sequence of a plant was Arabidopsis thaliana, and that was in the year 2000. So we're not that far from there. I might be showing my age to think that 23 years ago wasn't that long ago, but pretty exciting stuff that we're still just on the edge of. And so that's a model plant. Arabidopsis is used in a lot of different plant experiments because the whole genome is mapped. It's pretty neat to be able to look at all these different genetic markers. They mapped it, and then they're still working on other things to map, but it's about 1/13 the size of the human genome. I also have a very short window from seed to senescence, which is like the full life cycle of a plant. It's about 50 days for Arabidopsis. So you can use it for a lot of different experiments. And so it's a really cool little plant. You wouldn't really think much about it. It looks like a little weed. And so if you look it up, you're like, oh my gosh, this plant is like famous. But you wouldn't really think that to have it on your desk or anything like that. And so when we're thinking about selection, which is the traditional way of plant breeding that our ancestors used, just looking for those desirable traits and then culling those with less desirable traits, that is one portion. And then the other one is this deliberate interbreeding, which I mentioned with Mendel's pea experiments. And so you're crossing closely or possibly distantly related individuals to try to link their desirable properties together. And so, for example, like a mildew-resistant pea could be crossed with a high-yielding pea, but the high-yielding pea gets mildew problems. So we're like, okay, well, we want a mildew-resistant pea, but we also want it to produce a lot of peas. So how can we get both of those good things into one plant? And then you would cross those two together, and then you would continue to grow those subsequent generations to see if that came true. Where is the dominance? Where is the recessive? And so how are we balancing those things out? Because you really want something that's going to be predictable. You don't want to have, randomly a bag of seed that, it didn't quite take in this bag. And so that farmer, has this mildew problem that the other farmers don't have. And so it's really important that we have, consistency in our experiments. And so further development is always needed, and it takes a lot of input. We're going to discuss 3 different stories today about plant breeding, and I hope that everyone has their lunch out. And if anyone's having corn, then we're sharing that today. So let's start off with corn. The ancestor of corn you would not recognize as corn. So teosinte is the relative of corn. They have linked to our modern cob, as you would say. But on the DNA level, they do look surprisingly alike. So they, could tell that from the DNA history. Okay, teosinte, that's probably where this originated from. They have the same number of chromosomes and a similar arrangement of genes. And teosinte can crossbreed with modern corn varieties to form hybrids that can reproduce naturally. So that's pretty cool. But the morphological differences, the phenotypical differences of these are extreme. So teosinte only has like 6 to 12 kernels, and they're on 2 little rows, and they're protected by this hard outside covering. Whereas our modern corn has a cob that may have as many as 20 rows or more with exposed kernels. And so it took a long time for botanists to actually link the two together. It was unknown for a long time what the original ancestor of corn was. And so, not until we got into the genetics of it, we're like, oh, I think it might be this thing. But you wouldn't, if you looked at them right next to each other, you really wouldn't think so. This is one of our biggest success stories of domestication. And so this was just the hard work of the labor of Native Americans and their expertise of agriculture and paying attention to these things that we are able to enjoy the corn that we have today. And so one of the first things that they probably selected for was getting rid of that hard coating. And so today it's been so diminished that the only time you might encounter it is like sometimes that when you eat corn, it gets a little something stuck in your teeth. That's that. And so it used to be much more formidable to try to get through that coating, and it was difficult to chew. It's on the plant because it will protect the seed during digestion. And so the plant liked to have it around, but it didn't make a lot of sense for people when they were eating it. And so we were able to select out of that. Also, the architecture. Teosinte is like a multi-eared, multi-stalked, with the little kind of seedlets at the top. And now the plant that we have is a single stalk with a single ear, typically. And so it's probably easier to harvest, especially now with machinery. We want those ears to all be at a certain height. We want them all to be ready at a certain time. So the industrialization of agriculture really has depended on successful breeding programs over time. And so that corn, and who knows where we're going to go with corn. We're still working on getting our starch, figured out. And there's different kinds of corn. There's field corn that we feed to animals. There's corn that we use for ethanol production. There's corn that we use, sweet corn that we all enjoy straight from the cob. So many uses of corn. And it's just been a really interesting history of that crop over time. Another one I want to talk about today is sunflowers. I know it's a little gloomy outside, but sunflowers are always pick-me-ups, and so it's nice to see their little shining faces. But that was not originally how that plant looked. And so about 3000 BC was the first records of domesticating Helianthus annuus, which is the scientific name for sunflower. And it has evolved dramatically over the last several thousand years, about 5,000 years. And now it has a single head, big flowers. A lot of the seeds have a rich, high-quality oil to them. And so Native Americans cultivated this flower from its original bushy, multi-headed form so that it could produce a larger set of seeds. And these were used for different purposes. So they would use it for flour, to make bread or cakes. They could roast the seeds, delicious, cracked or eaten whole, mixed with other grains, kind of make like a granola. And then they were using this as a staple crop in several different areas also of the country. And so it's amazing the different types of sunflower seeds you can find now. Some of them are used for dye, some of them are used for oil, some of them are used for flour, and they all are have been selectively bred for these different purposes. And so it was a staple of North American residents for a long time, and then discovered by European explorers in 1510. The Spanish sailors gathered up large quantities of sunflower seed and shipped it back to Europe. But for about 200 years, Europeans didn't consider the edible qualities. They wanted them to be kind of this exotic showpiece in their gardens. And so they really didn't dive into diversifying that crop much. However, this, kind of just beautiful, sunshiny flower was being spread around, and during the 1700s had spread across Europe into Russia and Ukraine by the turn of the 19th century. So in Russia, the potential for oil production was established, and it wasn't necessarily because someone finally was like, we should use this for oil. And when I'm talking about oil, I'm talking about like a cooking oil kind of thing. We're not like putting it into our cars yet, as maybe, I don't know. But it was kind of a weird twist of fate. So the Russian Orthodox Church in the 18th century issued a declaration that for the period of Lent, they would ban the consumption of certain foods made from various oils and fats. And to help their parishioners, their churchgoers, adhere to this rule, they published a list of banned substances. But they did not have sunflower oil on that list. And so the demand for sunflower oil boomed, and the crop area expanded to more than 800,000 hectares across Russia and Ukraine in the early 1800s. The national flower of Ukraine is the sunflower. So I found that to be quite interesting. That acreage continued to grow, and the market for the crop diverged into two distinct areas: oil seed and seed consumption. And so as the consumer base was just demanding, different things, they started the first research programs in Russia to develop particular varieties to meet the consumer demand. And so I think that is pretty cool. So we have a large variety of sunflower called the Mammoth Russian, which was developed in the late 1800s, packed with hundreds of seeds, and they can be more than 50 centimeters in diameter. So they're pretty cool. These are the ones that I like to plant, and then I find in the summer they're so heavy that they're just like falling over. But it's a pretty cool story, and that's all because of plant breeding and just paying attention to what those traits were and understanding a little bit about genetics. All right, so we are going to talk about one more, and it is very seasonally appropriate. Poinsettias. So I don't have one in the office with me today, but it's that traditional Christmas plant that you've probably seen everywhere. It has red leaves, bracts, that surround kind of inconspicuous flowers, honestly. But the vibrancy of these red bracts, which look like leaves when you look up close, are just gorgeous, especially this time of year. It's so nice to brighten up something. I used to work at a bank in Colorado, and they actually had rules against plants in the bank. They wanted it to be a sterile environment. And so someone brought a poinsettia in for the holidays, and I wouldn't let them take it out. So we kept that poinsettia alive for months. I think it was like April before someone was finally like, oh, and I'm like, all right, I'll take it home. But it was really fun to be able to keep this plant in the office. So the poinsettia is a member of the Euphorbiaceae family. It was discovered in southern Mexico. So I got my Mexican corn casserole, hitting it up twice there. And we use it as an indoor plant. But in Mexico, they grow to be large woody shrubs, sometimes reaching 10 feet tall. So this is not the little compact thing that we know today. So how did it get there? Through plant breeding and different experiments. The ancient Aztec Indians of Mexico cultivated and regarded the plant as a symbol of purity. I cannot pronounce the name. I'm going to try. Quetzalcoatl. No, I'm sorry. I can't do that. Cuetlaxochitl, which means the mortal flower that perishes and withers like all that is pure. So it's just a beautiful translation. And so I will work on my pronunciation. I did look it up before this, and I thought I would be able to pull it off, but I need to practice. The Aztecs used this plant in many ways. They were making dyes out of the red bracts used in textiles and cosmetics. They crushed the plant for skin infections or used it to stimulate circulation. They made a medicine to treat fevers from the sap, which contains latex. And primarily we use it as an ornamental plant. So I couldn't find any current examples of people using this plant medicinally. They were introduced to the United States by the first appointed U.S. ambassador to Mexico in 1825. And he sent some plants to his home in Greenville, South Carolina. Joel Roberts Poinsett was his name. So where'd we come up with the name? And so that's in reference to him. And the poinsettias that we know today were really developed by the Ecke family. And so Robert and Paul began growing poinsettias in Hollywood and Beverly Hills. And the ranch is located in Encinitas, California now, and the major producer of all poinsettia mother plants used for cuttings by commercial growers. So about 70% of the poinsettias that we see today come from the Ecke Ranch in California. And it very slightly resembles its original parent. Like I said, the parent is leggy, tall, and so now we have it very compact. Typically that was created from, different trials of breeding, but also using cuttings, which creates this genetically identical material to the parent. And so we're going to talk in future sessions about asexual propagation and why that has been important for the plant breeding industry. But during the mid-1950s, people went really, they went for it. And so that's why we have like the whites and the creams, all these different colors. When you see one that has glitter on it, that is just paint. So occasionally they will put the plant in a dye and it will take up the dye. And so you can really see some interesting experiments on poinsettias. The reason why that we use them this time of year, though, is that they do put together their flowering phase during a shorter day cycle. So in order to get the poinsettias to do their thing and turn their bracts red, they really need 12 hours of darkness. And so greenhouses that are growing these will cover the plants to ensure that they don't get any random lights turned on. It's very important that they have this dark cycle. And so that's why they are a great plant for us this time of year, is because that's when we are getting that different light cycle availability. And so I won't see you guys until the 15th, but I did want to say that on December 12th, it has been declared National Poinsettia Day. So if you want to give someone a poinsettia on the 12th or before, you can participate in that. The last little piece I'll share with you about the poinsettia today ties into our pathology discussion. And so in the mid-90s, American plant pathologist Dr. Lee of the United States Department of Agriculture determined that a phytoplasma infection of poinsettias resulted in more compact plants with a greater number of flowers. And so phytoplasmas are a type of bacteria that affect the growth pattern of infected plants. And so the growers in the Ecke family had long utilized special grafting techniques to produce these compacted plants. But now we have widespread commercial adaptation of Lee's method, which is a lot of the result of what you'll see in stores today. So they introduced an infection to the plant that made it more compact and have more flowers, which, I mean, it's just incredible. So a lot of thanks and gratitude to the people out there trying new things. And, every success is surrounded by multiple failures. So these people didn't give up. And so I definitely recommend that if you want to try some little breeding projects of your own, you can set those up in your house. You can do some experiments with different plants that you enjoy, and tell us a little bit about some unique plants that you like in your life.